Good day, Mom. So my topic is all about the cellobiose. So cellobiose is produced as an intermediate in the hydrolysis of the polysaccharide cellulose. Like maltose, cellobiose contains two D-glucose monosaccharide units, but it differs from maltose in that one of the D-glucose units, the one functioning as a hemiacetal, must be a P configuration. So it is important to distinguish between structural notation used for an A1 to 4 carbon glycosidic linkage and that used for B1 to 4 glycosidic linkage. So we have here the configuration of the A and B glycosidic linkage. So in A, the carbon 1 and carbon 4 are balanced or they are in the same line, but in the P, the carbon 1 and carbon 4 are going up. So instead of a B configuration for maltose, this change in configuration results in a P glycosidic linkage. So cellobiose is reducing sugar as three isomeric forms in a aqueous solution and upon hydrolysis produce two D-glucose molecules. Cellobiose is a disaccharide composed of two glucose molecules linked by P glycosidic bond. It is an intermediate product formed during the breakdown of cellulose, a major structural component in a plant cell walls. Unlike maltose, which has an A glycosidic bonds, cellulobiose linkage gives it distinct biochemical properties. So this configuration shows the differences between maltose and cellulobiose. So maltose A linkage is easily recognized and broken down by enzymes like maltase, which humans and yeast naturally produce. This means maltose can be readily digested and utilized as a source of energy. On the other hand, cellulobiose B linkage requires an enzyme called cellulobase, which humans and yeast lack. As a result, cellulobiose passes through these systems without being broken down or absorbed for energy. So this difference is particularly important in nature. Like for example, cellulobiose is building block of cellulose, the structural materials in plant cell walls, and it plays a crucial role in the ecosystem where cellulose degrading organisms like certain fungi and bacteria to produce necessary enzymes to break it down. So while both maltose and cellulobiose are disaccharide of glucose, their glycosidic linkages define their digestibility, biological function, and in the role and in the and role in the environment. So like maltose, cellulobiose have also a formula. So D cellulobiose plus H2O H plus or cellulose and 2D-glucose. So despite these similarities, maltose and cellulobiose have a different biochemical behaviors. So these differences are related to the stereochemistry of the glycosidic linkages. Maltase, the enzymes that break the glucose A 1 to 4 linkage present in maltose. So is found both in human body and in yeast. Consequently, Maltose is digested easily by humans and readily fermented by yeast. Both human body and yeast lack the enzyme celluloase needed to break the glucose B linkages are of cellobiose. Thus, cellobiose cannot be digested by human or fermented by yeast. Humans and yeast lack the enzyme celluloase needed to break down this B bond or one 4 carbon bond, making cellulose indigestible for humans and unfermented by yeast. However, it can be hydrolyzed into 2 glucose molecules through enzymatic or acid hydrolysis. Celluloase is also a reducing sugar, meaning it can act as a reducing agent in chemical reactions.